In this module, we will see how the experiments of geneticists of Morgan allowed geneticists to make chromosomal maps. That is basically relative positions of genes along the length of a chromosome. I have shown you this slide before too. Now we are going to focus on a different aspect of this experiment. Here you can see we have 206 and 185 recombinant phenotypes. So what is the factor that determines the frequency of recombinant phenotypes? So if we wanted to calculate what is the frequency, it is a simple formula. Let's look at that. We basically add the number of recombinants and divide it by the total number of offsprings. And that gives us recombination frequency. Recombination frequency, we can have units for recombination frequency. We can convert this frequency into map units, and recombinant frequency of 0.01 equals 1 centimorgan. So if we do experiments and we see the recombination frequency of different genes along the length of chromosome, we can assign them relative positions. In this example, we have Y. You can see the Y is the body color right here. White eye is W. Vermilion eye is V. Miniature wings is M. And rudimentary wings is R. So during meiosis, these tetrads, these homologous chromosomes, when they come together, they form these tetrads. How frequently are these genes exchanged between these two homologous chromosomes? The further apart two loci or the locations of these genes, recombinant frequencies will be greater. So basically, if the distance between the two genes increases, so does the probability of recombination between these two locations. So using this information, scientists were able to make genetic maps which were basically relative positions of genes on a chromosome. Now let's take a hypothetical example. We have an organism with A, B, and C genes. We cross this organism and determine the frequency, the recombinant frequencies. First of all, we look at the recombinant frequencies between A and B genes. Here we have the data. So during this cross, we got 450 A, capital A, capital B, small b, and 450 small a, small b, small b. These are the parental phenotypes. The recombinant phenotypes are 50 capital A, small b, and 50 also of small b, and capital B, small b. These are the recombinant phenotypes. So the frequency, the way we will calculate their frequency is, as I have mentioned, we will add the recombinant phenotypes, which is 50 plus 50, and divide it by the total number of offsprings, which is 450 plus 450 plus 50 plus 50, the number equals 1000. So the recombinant frequency is 0.1. As we have already established that centimorgan, 1 centimorgan is 0.01. This is how you convert combination frequencies into centimorgans. So we can conveniently place the A and B genes along a chromosome, and the distance between them is 10 centimorgans. Next we do an experiment. Now we are looking at recombination frequencies between A and C. So here's the cross. These are the parents. We do this cross and we get the following data. We have 460 capital A, capital C, small c, and also the other parent type 460 small a, small c, small c. Here we get 40 recombinants of capital A, small c, and 40 recombinants small a, capital C, small c. Again, we use our previous exercise and calculate the recombinant frequencies. Here's the algebra. So we come up with the number 0.08. This number translates to 8 centimorgan. So we have now established the distance between A and C and A and B. What we don't know is the distance between C and B. As you can see, we can have different arrangements. You can see it on your screen in this region. We can have A, B and C, or A, C, B, or B, A, C. So we need to do one more experiment in which we will have to figure out distance between the C and B to complete our genetic map in this case. So let's do that experiment. So how far are B and C genes? We do the same cross, similar type of cross we have done previously. Now our recombination frequency is, the number of recombinants is 10 for capital B, small c, and 10 also for small b, capital C, small c. We convert this number into frequency. It is 0.02 or 2 centimorgans. Now with this information, we know the precise relative position of these three genes. A and B are 10 centimorgans apart, and A and C are 8 centimorgans apart, and C and B are 2 centimeters apart. This is how geneticists were able to make genetic maps for different organisms, and this information was very useful in it had practical applications also. You could predict what would be the combination frequency between, for example, two genes present on the same chromosome. Next, we will talk about the genetic information that we have learned. We will also talk about how this applies to humans in the next module.